Welcome! Let us learn how to use Scikit-learn to classify cancer cells as benign or malignant. Cancer cell classification with Scikit-learn. In this lesson, you will learn how to build a simple machine learning model to classify cancer cells. Accurate classification helps doctors detect if a cell is benign, not cancer, or malignant, cancer. Machine learning can help uncover useful signals in complex datasets with many measurements. You do not need any experience to follow along. We will use a popular dataset built into Scikit-learn, making it safe and easy. At the end, you will know how to train a model and check how well it performs. Running the cell now. Let us understand the idea behind classification, especially for cancer detection. What is cancer cell classification? Classification means teaching a computer program to assign a label, like benign or malignant, to examples based on their features. For cancer cell data, the model looks at measurements from cell samples and learns what patterns match each label. Good classification can save lives by helping with early detection and reducing human error. Running the cell now. Let us begin by turning off warning messages so our results are easier to read. Always start by suppressing warnings for a cleaner notebook. Import warnings. Import numpy as np. We set the random seed to 42 for reproducibility. Warnings.filter warnings, ignore. Running the cell now. This initial code sets up the environment by suppressing warnings and fixing the random seed for reproducibility, ensuring consistent results in later steps. Although no visible output appears, these preparations are crucial for clean, reliable analysis as we move forward with cancer cell classification. Suppressed warnings will help keep our focus on learning, not on cryptic messages. Now, let us load a built-in cancer dataset from Scikit-learn. This dataset contains features measured for many cell samples, and a label for each sample. We split the columns into featured data, X, and target labels, Y. Data setup, breast cancer dataset from Scikit-learn. From scikit-learn.datasets import load_breast_cancer. Data equals load_breast_cancer. X, Y equals data.data, data.target. Print, feature matrix shape, x.shape. Print, target label shape, y.shape. Print, first 5 labels, y5. Print, first 5 samples. Print, x5. Running the cell now. This output shows the shape and preview of the breast cancer dataset features and labels, confirming we have 569 samples with 30 features each, and a corresponding label for every sample. Notice the first 5 labels are all zeros, indicating they are benign cases, and the feature values are a mix of measurements like tumor size and texture. This setup is crucial as it prepares the data structure that a classification model will learn from to distinguish between benign and malignant cells. The feature matrix lists the measurements for each cell sample. Target labels show us if the cell is benign, 0, or malignant, 1. With this, we are ready to explore. Let us check what the target numbers and feature names mean. The target labels are 0 for malignant and 1 for benign. Print, target names, data.target_names. Print, feature names, data.feature_names. Print, number of samples, len, y. Running the cell now. This output confirms that our classification labels are a malignant, 0, and a benign, 1, providing clear categories for the model to learn. It also lists 30 detailed cell features, such as mean radius and texture error, which the model uses to identify patterns differentiating cancer cells. Finally, we see there are 569 samples, ensuring the dataset is large enough to train a reliable classifier. Malignant means cancer is present. Benign means not cancer. Our data contains many features, which are measurements taken from the cell samples. Knowing the sample count lets us see if we have enough data for learning. Good models need to be tested on new data. That is why we split data into training and testing sets. Splitting your data for training and testing. To measure how well our machine learning model learns, we split our data into two sets. Training data is used by the model to learn, while testing data checks how well it performs on new examples. This split helps us avoid overfitting, which is when a model memorizes everything but cannot generalize. Running the cell now. We will keep 80% of the samples for training and 20% for testing. Setting random_state makes your results repeatable. Let us split our dataset, 80% for training, 20% for testing. From Scikit-learn.model_selection import train_test_split. X_train, X_test, Y_train, Y_test equals train_test_split. X, Y, test_size equals 0.2, random_state equals 42. Print, training samples, X_train.shape, 0. Print, testing samples, X_test.shape, 0. Running the cell now. The output shows the dataset has been split into 455 training samples and 114 testing samples, reflecting an 80-20 split as intended. This confirms our code correctly divided the data, ensuring the model learns from the majority while being evaluated on a separate, smaller set to check for overfitting and generalization. Now we have a training set and a testing set. Having separate samples for testing gives us an honest measure of performance. Before training, check if both groups, malignant and benign, are represented in both sets. Balanced data means each class appears often enough to learn from. Data check, see if the classes are balanced in both sets. Import numpy as np. Print, training set label counts, np.binCount, y_train. Print, testing set label counts, np.binCount, y_test. Running the cell now. This output shows the count of each class label in both the training and testing sets, highlighting that the split maintains a similar balance between classes. Notice that both sets have proportionate numbers for each label, which is crucial to ensure the model learns and is evaluated fairly across all categories. This check confirms that our data splitting step preserves class distribution, helping avoid biased performance estimates. If any class is missing, the model cannot learn about it. Both should be there in training and test data. Since measurements are in different units and ranges, standardizing, scaling them helps the model. We use standard scaler so features have mean 0 and unit variance. Optional, standardize features so all measurements are on a similar scale. From Scikit-learn.preprocessing import standard scaler. Scalar equals standard scalar. X_train_scaled equals scalar.fit_transform X_train. X_test_scaled equals scalar.transform X_test. Running the cell now. This code doesn't produce visible output but is critical because it standardizes the training and testing features to the same scale. Notice how we fit the scalar only on the training data to avoid leaking information, then apply it to both sets, ensuring our model learns on consistent data. This step supports fair evaluation by preventing scale differences from skewing model performance during testing. Now our features are in a similar range, making learning smoother for most models. Next, let us decide which machine learning method to use. Choosing a machine learning algorithm. There are many algorithms for classification. We will use logistic regression, which is simple and easy to interpret. Logistic regression is commonly used for binary classification. It predicts the chance that a cell is malignant or benign. We will use Scikit-learn's built-in implementation. Running the cell now. Let us train our logistic regression model using the pre-processed training data. Max_iter means the model will try enough times to find a good solution. Train a logistic regression classifier. From Scikit-learn.linear_model import logistic regression. CLF equals logistic regression, max_iter equals 10,000, random_state equals 42. CLF fit, x_train_scaled, y_train. Running the cell now. This output confirms that the logistic regression model has been successfully initialized and trained with a maximum of 10,000 iterations to ensure convergence. Notice the use of random_state equals 42, which makes the results reproducible. This step is key as it prepares the model to predict whether a cell is malignant or benign, aligning with our goal of applying a simple yet effective binary classification algorithm. Our model is now trained. It has learned to look for patterns that separate malignant and benign cells. Now, let us check how well the model predicts for both seen, training, and unseen, test, data. Accuracy is the fraction of correct guesses. Check the accuracy of the model on training and test data. Train_accuracy equals CLF score, x_train_scaled, y_train. Test_accuracy equals CLF score, x_test_scaled, y_test. Print, gef training accuracy, train_accuracy a 0.2f. Print, f test accuracy, test_accuracy a 0.2f. Running the cell now. The output shows that our logistic regression model achieves very high accuracy, with 99% on training data and 97% on test data. This indicates the model is learning well and generalizing effectively to new, unseen samples. These accuracy scores help us evaluate if logistic regression is a good choice for classifying malignant versus benign cells in this dataset. If training accuracy is much greater than test accuracy, the model may be overfitting. In general, high accuracy is a good sign, but test accuracy is what really matters. Accuracy is only one measure. A confusion matrix shows us where the model made mistakes. A classification report shows precision, recall, and F1 score, which explain different aspects of performance. Look deeper, show a confusion matrix and a classification report. From Scikit-learn.metrics import confusion_matrix, classification_report. Y_pred equals CLF predict, x_test_scaled. CM equals confusion_matrix, y_test, y_pred. CR equals classification_report, y_test, y_pred, target_names equals data.target_names. Print, confusion matrix \n, cm. Print, \n classification report o \n, cr. Running the cell now. This output shows how well our logistic regression model classifies malignant and benign cells. Notice the confusion matrix has very few misclassifications, 2 false positives and 1 false negative, indicating strong performance. The classification report confirms this with high precision, recall, and F1 scores above 0.95 for both classes, demonstrating the model's ability to accurately predict cell diagnosis in this binary classification task. Precision measures how many positives predicted are correct. Recall measures how many true positives the model finds. F1 score is a balance between precision and recall. These help us understand not just if the model is correct, but how it makes mistakes. Let us plot the first 2 features to see if the data can be separated visually. Each color shows a different class. Visualize, plot a few features to understand separation. Import matplotlib.pyplot as plt. plt.figure, figsize sad smiley 7.5. plt.scatter, x_train_scaled, 0, x_train_scaled, 1, c equals y_train, kmap equals coolwarm, alpha equals 0.6. plt.xlabel, data.feature_names, 0. plt.ilabel, data.feature_names, 1. plt.title, cancer cell samples, first 2 features. plt.show. Running the cell now. This scatter plot shows the distribution of cancer cell samples using the first two scaled features, colored by their labels, malignant versus benign. Notice how the different classes form somewhat overlapping clusters but still show some separation, which is important for logistic regression to find a decision boundary. This visualization helps us understand how the features might contribute to the model's ability to classify the cells. If you see clusters by color, it suggests the features help the model learn to separate classes. Machine learning finds boundaries in many dimensions, not just two. Let us see how easy it is to test a different classifier. Trying other models, support vector machine. You can try other classifiers, like support vector machine, SVM. SVM tries to find the best boundary that separates the two classes. Changing algorithms helps you see which method works best for your data. Running the cell now. Now we use a linear SVM. SVMs work very well when data can be separated by a line, or plane, in some feature space. Let us compare its accuracy to logistic regression. Train and test a support vector machine, SVM, classifier. From Scikit-learn.svm import svc. svm_model equals svc, kernel equals a linear, random_state equals 42. svm_model.fit, x_train_scaled, y_train. svm_test_acc equals svm_model.score, x_test_scaled, y_test. Print, f svm test accuracy, svm_test_acc 0.2f. Running the cell now. The output shows that the support vector machine, SVM, classifier achieved a test accuracy of 96%, indicating it did a great job separating the classes with a linear boundary. This high accuracy highlights how trying different algorithms, like SVM, can help identify which model best fits your data. Notice that this approach complements previous models by possibly providing better or comparable performance. If the SVM accuracy is similar or better, you might choose this for some problems. Always compare different models to see what works best. Logistic regression can tell us which input features matter most for its predictions. A large value, positive or negative, suggests the feature has high importance. Bonus, feature importance with logistic regression coefficients. Import pandas as PD. Coef equals CLF coef_0. Importance equals PD series, coef, index equals data.feature_names. Print, importance.sort_values, key equals abs, ascending equals false, head, 10. Running the cell now. This output lists the top 10 most important features based on logistic regression coefficients, showing their impact on the prediction. Notice that all these features have negative coefficients, meaning as their values increase, the model is more likely to predict the negative class. While this helps interpret logistic regression, it sets the stage for comparing how SVM might weigh features differently when finding the best class boundary. You now have some insight into which characteristics of the cancer cells mattered most to the model. Doctors can learn from this, too. Let us see how our model predicts the class of just one cell sample. We will use the first example in the test set. Make a prediction for a single sample from the test set. Sample equals x_test_scaled, 0. True_label equals y_test, 0. Predicted_label equals CLF predict, sample, 0. Print, f true label, data.target_names, true_label. Print, f predicted label, data.target_names, predicted_label. Running the cell now. Here, the code predicts the class of a single test sample using the trained SVM model. The output shows that the true label is benign and the predicted label is also benign, indicating the model correctly classified this example. This highlights how SVM can effectively separate classes by finding a good decision boundary. Try your own input samples, or change the index to see results for different cells. Seeing a single case helps us connect the model's output to the data. Here, we simulate a new patient by picking the average feature values and adding a little random noise. Try running this cell a few times and see how the predictions may change. You could replace the values with your own made-up feature list to see what class your data would belong to. # try, predict for a new, simulated, sample. Import numpy as np. User_sample equals np.mean, x_train, axis equals 0, plus np.random.random, x_train.shape, 1, asterisk 0.1. User_sample_scaled equals scalar.transform, user_sample. Prediction equals CLF predict, user_sample_scaled, 0. Print f predicted label for your made-up sample, data.target_names, prediction. Running the cell now. Here, the code predicts the class of a new, simulated sample using the trained SVM model. Notice that the prediction is malignant, showing how the model applies the decision boundary it learned to classify unseen data. This connects to the lesson's goal of testing how well SVM can generalize beyond the training set. Experiment with changing values to see how the model responds. That is how scientists learn, too. Let us have some fun. Please enter 5 cell measurements below. If you are not sure, you can make up numbers similar to what you saw in the data samples. We will fill out the remaining features with average values, to keep it simple. #mini project, ask the user for measurements and predict cancer cell type. User_features equals. For name in data.feature_names, 5. Val equals float, input, f enter value for a name. User_features.append, val. User_features equals user_features plus list, np.mean, x_train, axis equals 0, 5, smiley face. User_features_scaled equals scalar.transform, user_features. Prediction equals CLF predict, user_features_scaled, 0. Print f prediction, data.target_names, prediction. Running the cell now. Here, the code takes user input for the first 5 features and fills in the rest with average values before scaling and predicting with the SVM model. Notice how this allows testing the model interactively on custom data, helping you see how the SVM classifier separates and labels samples as malignant or benign. This step shows the practical use of SVM in real-time predictions based on feature values. Great job trying it out! This is a small taste of how real applications work. Remember, always check results with a real doctor. Machine learning gives clues but is not a replacement for experts. Let us summarize what you achieved. What you learned. You loaded and explored a real-life cancer dataset. You prepared your data for learning and visualized it. You built and evaluated a classifier using Scikit-learn. You checked model performance using several metrics. You even tried a small project with your own values. Try substituting other Scikit-learn datasets and algorithms to keep learning. Subscribe to our YouTube channel for more beginner-friendly data science lessons. Running the cell now. Congratulations on completing your first cancer cell classification project. Keep practicing and share your results with others.